In a Virginia forest near Johns Creek, a scientist crouches beside a young chestnut tree. She doesn't reach for a shovel. She reaches for a printout of its DNA. On that page, in 4 letters repeated thousands of times, is the answer to whether this tree will live or die from a plague that killed 4 billion others. The tree everyone bet would save this species was engineered in a lab, hyped in every newspaper, and funded with millions. It collapsed. And the thing actually bringing the chestnut back is older, slower, and stranger than anyone planned. Here's exactly how. To understand why that page matters, you have to understand what was lost. And here's the strange part. Almost nobody alive has ever seen it. Go back 150 years and walk into the forests of Appalachia. Roughly 1 in every 4 large trees is an American chestnut. They stretch from Maine to Mississippi across something like 200 million acres. Some are 10 stories tall. In autumn, they drop so many nuts the ground looks like it's covered in snow. Bears eat them, deer eat them, wild turkeys eat them. Farmers turn their pigs loose under the branches and let the forest fatten them for free. The wood barely rots, so people build cabins, fences, cradles, and coffins out of it, cradle to grave, all chestnut. This one tree is food, money, and shelter for an entire mountain culture. Now look at the same forest today. The chestnut is basically gone. Here's the mystery. It didn't fully disappear. If you hike those hills right now, you'll still find chestnuts, but they're pathetic. Skinny sprouts, maybe 15 feet tall, growing out of ancient stumps, covered in ugly orange cankers, dying before they can flower. People walk past them and think that's just what a chestnut is. A scrubby little understory shrub. That's the first thing you have to unlearn. What we call a chestnut today is a ghost. A sick, stunted echo of a giant that most people simply forgot ever existed. The baseline itself is broken, and we mistook the broken version for normal. So what happened? In 1904, a gardener at the Bronx Zoo noticed orange spots on the bark of the chestnut trees and leaves that were curling and dying. The culprit was a fungus, Cryphonectria parasitica, that had hitched a ride into the country on ornamental chestnut trees imported from Asia. And this fungus had a brutal method. It slips into a wound in the bark, grows underneath, and pumps out oxalic acid. That acid eats a wound into the trunk called a canker. The canker spreads around the trunk like a belt tightening, and once it wraps all the way around, it strangles everything above it. Water and nutrients can't get past. The whole crown dies. Then comes the cruel part. The roots don't die. They send up a new sprout. The sprout grows a few years, gets infected, gets strangled, dies back to the root, and again. And again. A living tree, trapped in a loop, never allowed to grow up. The walking dead of the forest. It spread like fire. By 1925, it had blown 1,000 miles out from New York in every direction. By 1950, an estimated 4 billion American chestnuts were dead. 4 billion. In under 50 years, the most common giant in the eastern forest was functionally extinct. And people tried to stop it. They really tried. They cut down infected trees, they burned them. They set up quarantines and cleared whole bands of forest hoping to build a firebreak the fungus couldn't cross. It failed. Completely. The fungus made spores by the millions, and they rode the wind, rode the rain, rode the feet of birds and insects. Worse, the fungus didn't even need chestnuts to survive, and it could live quietly on oaks and other trees, waiting. You cannot quarantine the wind. Every attempt to attack the fungus head-on lost. So for decades, the chestnut just sat there in that dying loop, and the story looked over. Then, generations later, biotechnology showed up and promised a silver bullet, and almost everyone believed it. That was the second mistake. Here's the idea that captured everyone. Scientists at the State University of New York's College of Environmental Science and Forestry asked a simple question. What if you don't fight the fungus at all? You just take away its weapon. Remember the oxalic acid? The stuff that eats the canker into the trunk? There's a gene in wheat that makes an enzyme called oxalate oxidase, and that enzyme breaks oxalic acid down into harmless byproducts. So the team inserted that one wheat gene into the chestnut's DNA. The plan was elegant. Don't kill the fungus. Just disarm its acid and let the tree wall off the infection and survive. They called their star tree Darling 58, and the hype was enormous. Darling 58 got written up in the New York Times, the Wall Street Journal, the Washington Post, National Geographic. It was pitched as a revolutionary transgenic tree, the first genetically engineered organism ever released to save a wild species. The American Chestnut Foundation, the non-profit that had spent decades on this fight, threw its weight behind it, pouring in something like $2 million in funding and roughly $2 million more in legal and scientific support. They filed a petition asking the US government to deregulate the tree so anyone could plant it in the forest. This was going to be it. The comeback. Everybody had waited a century for. Then, on a Friday evening in December 2023, the foundation quietly put out a press release and pulled the plug. Wait, what? After all that money, all those headlines, all those years, the foundation announced it was withdrawing support for Darling 58, and the reasons were rough. In field trials, the engineered trees grew slower and shorter than expected. Their blight resistance was inconsistent. Sometimes it just didn't hold. Survival was worse than promised. And then came the detail that turned a disappointment into a genuine scandal. The trees they'd been calling Darling 58 for years weren't Darling 58. Somewhere back around 2016, during routine work moving plants for pollination, the lines got mislabeled. The tree that had been studied, promoted, and pushed toward government approval as Darling 58 was actually a different line, Darling 54. And Darling 54 had a hidden flaw. The insertion had accidentally disrupted one of the tree's own genes, a gene called SAL1, which may have quietly sabotaged the tree's health. For something like 7 years, the flagship of the entire genetic engineering effort had been the wrong tree. Andrew Newhouse, who directed the project, admitted it plainly. They were only now understanding that the trees they'd been working with since 2016, the ones they'd been calling Darling 58, were really Darling 54. And in the fallout, Newhouse said something that cut deeper than the labeling error. Biotechnology, he acknowledged, probably wasn't going to be the best tool to deal with all of these threats. The silver bullet was a misfire. But step back, because the mislabel is not actually the deepest lesson here. Even if every label had been perfect, there was a bigger problem hiding underneath the whole approach, and it is the key to the entire story. One gene was never going to be enough. Which raises the question nobody had truly answered in 100 years of trying. If it isn't one gene, what actually stops the blight? To answer that, look at the tree that survives the fungus just fine. The Chinese chestnut. Chinese chestnuts grew up in Asia alongside this fungus for millions of years. They evolved with it. And when the blight hits them, they do not die, they shrug it off. So for most of the last century, the plan was obvious. Cross the American chestnut with the Chinese chestnut to borrow that resistance, then breed the offspring back to American chestnuts over and over. Backcrossing, it is called. Keep the resistance, but wash out the Chinese traits until you have got a tree that is almost entirely American, tall, fast, forest-shaped, but carrying the Asian shield. Simple, right? So why did it take a century and still not finish? Because of what the newest research finally proved. In 2026, a team led by Virginia Tech and the American Chestnut Foundation, working with the Hudson Alpha Institute, Oak Ridge National Laboratory, and the U.S. Forest Service, published a study in the journal Science that cracked open how chestnut resistance actually works. It is not one gene. It is not a switch you can flip. Resistance is spread across many genes, scattered all over the genome, all working together, thickening cell walls, triggering chemical defenses, slowing the fungus down from a dozen directions at once. It is not a lock with one key. It is an entire immune system built over millions of years. And suddenly, the Darling 58 failure makes total sense. The engineers tried to disarm one weapon. The tree needed a whole army. You cannot bolt one wheat gene onto a chestnut and reproduce what evolution spent millions of years assembling. So breeding it in was the right instinct all along. But breeding ran straight into a trap, and this trap is the real reason the chestnut stayed lost for so long. Here is the trap. If resistance is scattered across dozens of genes, then every seedling from a cross is a genetic lottery ticket. Some inherit a great combination. Most inherit a mediocre one. As the lead scientist, Jared Westbrook, put it, each American chestnut hybrid tree is a roll of the genetic dice. And to find out which seedlings won the roll, you had only one option. Infect them with the blight and wait. Wait years to see which ones survived, and most of them did not. A chestnut can live over 100 years, so every single breeding cycle was a decade-long gamble played half-blind. Think about how bleak that is as a job. You raise a tree for years, you deliberately stab blight fungus into its bark, and then you watch to see if it dies. Do that across thousands of trees, across decades, and you start to understand why 100 years went by with the chestnut still stuck as a shrub. That was the real enemy. Not money, not effort, time and invisibility. For 100 years, breeders were working in the dark, unable to see which trees carried the shield until the trees were old enough to be tested nearly to death. They were breeding blind. So how do you fix that? Not by building a fancier tree, by finally turning on the lights. That's what the 2026 study did. The researchers sequenced the DNA of thousands of chestnut trees that had already been through years of blight testing. They matched genetic patterns against real-world survival, who lived, who died, and why. Once you know which genetic signatures go with resistance, you don't have to infect a tree and wait anymore. You can read a seedling's DNA and predict whether it'll survive while it's still tiny. They call it genomic selection. Jason Holliday, the Virginia Tech geneticist on the study, put it simply, "This work changes how fast we can move. Instead of waiting years to see how a tree performs, we can use its DNA to predict resistance and make better decisions much earlier." It sounds like a spreadsheet trick. Then they ran the numbers on the next generation. With DNA-guided breeding, the team projects the next generation of trees will have about twice the average blight resistance of the current population, while still being, on average, about 75% American chestnut. Sit with that number for a second. 75% American means the tree keeps the things that made the chestnut matter: the height, the speed, the ability to shoot up through the forest and fight for the canopy instead of sulking in the shade like the Chinese chestnut does. It's not a foreign tree wearing an American name. It's mostly the real thing, carrying just enough borrowed immunity to survive. And here's the reversal at the heart of all of it. Nobody engineered that resistance. The tree already had access to it, through its Asian cousin, through its own genome, through the slow logic of breeding. The scientists didn't build the defense, they just learned to read it and to pick the seedlings that already carried it, years before the blight could ever touch them. They didn't invent a new chestnut, they stopped breeding blind. Now, you might be pushing back already. 75% American, is that even still an American chestnut, or is it some hybrid pretending to be one? It's a fair question, but think about what that quarter of Chinese ancestry is actually doing. It's not reshaping the tree, it's carrying the immune genes and almost nothing else, while the American three-quarters keeps the trunk, the height, the growth, the shape of the tree people lost. Close enough that in a forest, you would simply call it a chestnut. Westbrook estimates the next generation of trees should start producing large amounts of seed for real forest restoration within the next decade. Not a lab curiosity. Seed for forests. And this isn't theoretical. Near Johns Creek, in the George Washington and Jefferson National Forest, there's a stand of backcross hybrids that were planted back in 2012. One of them, now more than 13 years old, stands tall and green while the fungus is all around it. Cassie Stark, a regional coordinator with the Chestnut Foundation, works with trees like it, living proof that a mostly American chestnut can take a hit from the blight and keep climbing. These are the trees whose DNA taught the computers what resistance looks like. But, and this matters, is it back? No, not yet. And this is where the story stays honest. Because there was one more hope people clung to, and the same study quietly tested it. Scattered across the eastern US are a handful of wild American chestnuts that somehow survived decades of blight, big, old survivors that shouldn't exist. People hoped these trees were secretly resistant, that the species had quietly saved itself, and we just had to find them. The researchers checked, and the answer was deflating. Those rare survivors pass on only modest, inconsistent resistance to their offspring. They can help. They cannot save the species on their own. The chestnut did not secretly fix itself in the woods while we weren't looking. So the comeback is real, but it's a head start, not a finish line. And then the story gets stranger. Because there's another way the blight has been beaten. Not in America, in Europe. When the same fungus invaded European chestnut forests, it started the same way, mass death. But then something odd happened. The forests started healing on their own. The cankers stopped strangling the trees and instead turned into shallow, harmless scars that the tree just walled off and grew past. The reason turned out to be almost science fiction. A virus. There's a virus called Cryphonectria. Hypovirus 1 that infects the fungus itself and makes it sick. A weakened fungus can't pump out enough acid to girdle a trunk. It just makes a superficial blemish. In Europe, that virus spread naturally through the fungus population, and chestnut stands came back across whole regions, essentially for free. Nature wrote a cure, aimed not at the tree, but at the tree's killer. So why not just bring that virus here and heal America's forests too? They tried, and with a few exceptions, like some pockets in Michigan, it flopped. Here's the physical reason. For that virus to spread, sick fungus has to fuse with healthy fungus and pass the infection along. But American populations of the blight fungus are wildly diverse, and they come in so many incompatible types that they refuse to fuse with each other. The virus hits a wall it can't cross. The same trick that healed a continent stalls out in American soil. Picture the two side by side. On one tree, an angry orange canker wrapped around the trunk like a noose. On another, in an Italian forest, the same fungus reduced to a flat, calloused scar. The tree simply grew over. Same fungus, same species of tree. Two completely different endings, decided by whether a virus could get from one strain to the next. Which tells you something the whole century keeps repeating. There was never one magic answer. Every fix, whether engineered gene, imported virus, or wild survivor, worked only where the conditions were right and failed where they weren't. And that's exactly why the DNA breeding approach is believable instead of hype. It isn't a miracle. It doesn't claim to work everywhere overnight. The trees are only about 75% American, not the original. Restoration seed is still a decade out. Nobody has proven you can scale this to the 200 million acres the chestnut once ruled, and the fungus is still out there, lurking on the oaks, patient as ever. This is not a victory lap. It's a door finally cracking open after being jammed shut for 100 years. So let's go back to that scientist crouched by the young tree near Johns Creek, holding a printout instead of a shovel. For a century, people tried to outmuscle this blight. They burned forests to stop it. They quarantined counties. They spliced a gene from wheat into a tree and put it on the front page. Every one of those was a bet that we could engineer our way out with something new. The tree never needed something new. It needed something old, the layered, many-gene defense that evolution already invented, carried back in the slow, unglamorous way, generation by patient generation. And it needed us to finally learn to read what was already written inside it. Holliday put the whole thing in one line. "We're not just learning why the chestnut was lost," he said. "We're learning how to bring it back." The comeback. Everyone's waited for isn't the silver bullet from the lab. It's the oldest idea in the story, the tree's own defenses read at last letter by letter, not newer engineering. Older engineering, finally understood. And if you like these, the stories where the answer everybody overlooked turns out to be the one that works, stick around. There are a lot more where the cheapest, oldest, strangest looking fix quietly won.